Yep, and I think here we go again. Hi, hello everyone. I'm Oliver, Microbehunter here. I'm happy that you can join me again in this Saturday's live stream. I think everything should be working. At least I can see myself on YouTube. And I think the audio should also work. Yeah, okay. And what you see here in the background already here on the side here, that's already a sample of kefir. And those tiny dots here are kefir bacteria and also yeasts. And today in this live stream, I want to do two things. I want to show you how I've concentrated those bacteria. So we'll be doing a little bit of a specimen preparation again. So I'll be using the centrifuge again. And then I also want to talk a little bit about phase contrast microscopy as such, a little bit some microscope hardware, because people wanted to know a little bit more about that. So I decided to do both of this here. I see that many people have already started to join the chat. Yeah, I would like to say that if you, quite happy that everything seems to be working. Okay. If you have any questions, please feel free to post it in the chat. I do also want a short organizational thing here. I'll do the live stream today will last approximately maybe an hour or so. Okay, so it's going to be a little bit shorter. And just for organizational reasons here. What you see here in the back here, those tiny dots here are actually bacteria, Kefir bacteria. And it's a probiotic dairy drink, and those are things, the large structure that's basically coagulated milk, protein, and so on, right? What I would like to do today is I would like to show you a preparation technique on how you can do that. If you have any questions or so, please feel free to post them, of course, as every week in the chat. And please also indicate maybe at Microbehunter or at Oliver, so it's easier for me to actually find the chat comments. So let's start at the very basics here. Maybe many of you already know what kefir is. For those of you who don't know, I've got over here a ready-made one. So I already made kefir myself at home, but I bought commercial kefir. Not sponsored. Okay, doesn't matter. But that is basically it looks like yogurt. Yeah, and in this case, it tastes actually also quite similar to yogurt. I put already some of this out here, and I like eating it, just like I like eating yogurt. And it is, of course, a microbiological product. What I would like to do, therefore, is I would like to show you first a short video on how I used to make kefir myself. Maybe some of you have also made it yourself as well, but today I'll be using this commercial kefir, which also works. Okay, so I'm going to quickly show you a video, and I'm going to narrate this because it's actually quite interesting. Because what you need is a kefir fungus, and we have this over here. So I bought myself a couple of years ago. So the clip I made already some time ago. I basically bought myself some kefir. It came in this little plastic bag. And I just want to show you now how the actual kefir grains, how they look like. Those kefir grains are a community of bacteria and also fungi, specifically certain yeasts. Okay, so that is what I've done. First, I pressed it out here, and then I rinsed it with a little bit of water simply to show you those kefir grains. And what you do is you then add some milk to those kefir grains, and you let it stand at room temperature, and then after several hours, it's going to make the kefir drink. Okay, so this is how, and then I just simply washed it with a little bit of water, and then you basically see those clumps that you see here. That's actually a community of yeasts and also bacteria. Okay, so what I'm going to probably do is I'm quickly going to pause the video over here, right? So that's how it looks like, and every time when you add milk to it and when you allow it to ferment, then they will actually also start to grow. Okay, so that's there's more and more. And then if you have a lot, then you give some of those to your friend, or you basically throw it away, or you sell it, right? But they will actually grow, and that's basically a community of yeasts, which are fungi and bacteria, and those bacteria, they produce a polysaccharide, and which is kind of this sticky matrix, and then you have this complex community of yeasts and bacteria growing in those clumps, those kefir clumps here. right? So, and basically, yeah, there are different types and depending on the type of taste that you want. Sometimes the kefir drink also tastes a little bit fizzly because of it's carbonated, because those yeasts, they produce carbon dioxide, also even a little bit of alcohol even. Others, like the commercial one that I have here, does not taste, it tastes more like yogurt. Okay, and not, yeah. So there are different types around there. And what I've done before is I put some of those kefir grains directly under the microscope, and you're not going to see a lot because it's also dense, and the cells are also densely packed together in this polysaccharide matrix. You're not going to see anything. So I was very disappointed. But luckily, many of those bacteria and yeasts, they also start to break off, and they start to actually then float around in the actual kefir drink itself. And this is where we can extract them. Okay, so this is something that I just want to show you now, and how I've done this extraction process, and this is basically the result here. So I've basically already today, a few hours ago, I've prepared this already simply as a backup in case it doesn't work. And the nice thing is that you see those tiny bacteria here in the background floating around. Okay, so, and we're gonna try to get there. So what I'm gonna do the first is I'm going to directly put some kefir under the microscope. Simply to show you how pure kefir looks like and why we have to do the centrifuging part. So I'm going to just take a toothpick and I just put a little bit of kefir directly on here. You don't want to use too much of this. Let's just put it back in here. Okay, let's make it a little bit bigger. Yeah, a tiny amount is enough. You put a cover glass, of course, on top. Yeah, and yeah, and every now and then, don't worry. I'll be also reading the comments, of course. Yeah, but let me just get started a little bit. I put a cover glass on top, and of course, you want to press it flat. Okay, so that's now pure kefir under the microscope, and let's just want to show you for comparison how it looks like. Okay, so that's a little bit the idea. I'm gonna put it again under the microscope here. Okay, so that's the one from before, and that is the one right now, directly the kefir, and you can already see the problem, right? And the problem is that all of this stuff here, that's coagulated milk protein, and it's so dense that I'm not able to see the bacteria, right? Of course, here in between, you're going to see a few of them, right? Occasionally, if you're lucky, maybe you see the odd one floating around here. But honestly, gee, where are they, right? Yeah, so evidently, yeah, here a little bit maybe. You know what, I'm going to switch on phase contrast. Maybe we see it a little bit better, but I think you already see it. Putting kefir directly under the microscope is not really, yeah, let's turn it up a little bit here. It's too dark even here. You see, no, nothing here. That's, yeah, even here with the bacterial concentration is too low. There is too much of this matrix, this coagulated milk protein. I don't see any bacteria, right? Yeah. In comparison, again, this is yeah, the other one from before, also now in phase contrast. You see those tiny dots in the background. Yeah, these are the bacteria. That's what I want to see. Yeah. So evidently, I have to somehow process over here maybe a couple floating, but the concentration is way too low, right? Yeah, so that's why we have to centrifuge this, okay? And I just want to show this to you. I hope that it works again. At least it worked 2 times today in the afternoon, and I just want to show this to you. And yeah, what I have to do, therefore, I have to now put the centrifuge directly on here on the table. Actually, I put the centrifuge on my floor with the second camera, but the problem is that the camera didn't work. Okay, so what I have to do now is I have to, yeah, do first a slight preparation over here. And what I'm going to do now is the following: is I'm going to put some of this kefir into this beaker over here, right? So that's just the regular commercial kefir, no kefir grains, obviously, because yeah. I'm just going to add some of this stuff here. Okay. Yeah, I think this should be kind of enough, or maybe a little more. I don't know. Yeah, and what I do now is yeah, there's a question: are the bacteria dead or dormant in the field? No, they should be alive. The bacteria should be alive. Maybe not all of them. Sure, certainly some of them might have died because they ran out of nutrients. But honestly, plenty of them should still be alive. right? So one way of testing it is actually trying to take this kefir and adding some milk to it and then see if it continues to ferment. And what I do is because this is too viscous, I add a little bit of, I don't know, maybe the same amount of water. Okay, and now just realized something that I completely forgot, that I actually need a spoon to stir it. Because I don't have a spoon, I'm just going to do it, I'm going to cheat a little bit. I'm going to use a spatula, super well prepared. So, and I'm just going to use this spatula here, and we're just going to mix this. Okay. So we have now, of course, a nice suspension of bacteria and kefir and a whole bunch of milk protein. And now I need to kind of separate this out. And in order to do that, I think it's properly mixed now. I have to centrifuge it, okay? And what I'm going to do therefore is the following, is I'm going to take myself a centrifuge tube. Okay, I labeled this one already. Okay, this one is a clean one here. Okay, here we go. This is just a regular plastic centrifuge tube. And I'm going to now add, a little bit up to here, approximately. I'm going to add some of this kefir suspension with bacteria. And we hope that this should be okay. Okay, you see that's looks actually like milk, right? I put a lid on top here. I put this on the side, and now it's going to become noisy. And now I have to do the following. I have to actually move the centrifuge into the center here a little bit more so that you see what I'm actually doing. Ah, look at this thing. I'm going to do it like this here. And this is going to be pretty noisy, okay? And I'm going to, over here, I have the speed setting. And what I'm going to do is I'm going to go at around, I don't know, intermediate setting. What does it say here? 4,000 is full. Maybe this is around 2,000 or so, yeah? Can kefir be eaten directly? You mean the fungus? Why not? Why not? It's certainly not poisonous, because if it were, then you wouldn't be able to drink, because you got the same bacteria anyway floating around in the kefir. So I see no reason. I don't know if it tastes good. Never tried it. So I'm going to put this in here. By the way, this is a counterweight, so it should be pretty much more or less the same. Yeah. And what I'm going to do now is, for about a minute or so, I'm going to spin it at a low speed, so that the solid material will actually be pulled down, okay? And it's gonna be loud now, okay? So I don't know if you're able to hear me, so if you wanna, I don't know, okay? I'm gonna switch this up. So this is now, it's starting to vibrate a little bit. Yeah, and I'm going to leave it at this speed setting for maybe a minute or so. So, and in the meantime, if you're still able to hear me, I don't know. I'm going to read a couple of your comments over here. Off-topic question: does anyone post their microscope images on iNaturalist? iNaturalist, highly recommended app on the mobile phone, but also can be done online. Yeah, make yourself a free account and start posting pictures of animals, plants, and of course also of microorganisms. I think it's a beautiful and a very nice app, which also does identification. So it has this AI system to actually also identify microorganisms. Yeah. The next question is aren't the bacteria dead or dormant in the finished kefir? I mean, they have done their job on the fresh milk, turning it into kefir, so they don't have much food left. That's correct. Generally, what happens is that this is called by microbiology, it's called a stationary culture. This means that the bacteria and yeasts, they stop dividing. They stop dividing, but they're still alive. Okay. And if you add more milk, then they will start dividing again. Yeah. Can we do a drop without centrifuge too and directly compare the difference? Yes, I did not read that comment before, but that's what I just did. Okay, so yeah. And can kefir be eaten directly? I see no reason why not. Okay, so I think this should be enough. Okay, so yeah. Okay, it's fine, not too loud, good. Yeah, so yes, here we go. And so this one is the counterweight, and over here, I hope we're able to see very nicely. Okay, we're able to see now the following. We're able to see that the top part, you see the quite solid over here, and here the top part is still cloudy. There should be a lot of bacteria still in here. So what I'm doing right now is I'm going to simply decant the whole thing and put it over into a new tube. Hope you don't see that. Okay. Basically like this. This one I put on the side, and now I have to add again a little bit of water to make sure that the counterbalance, and I'm going to centrifuge it again. You have to be always careful that you're actually able to see this. Up here we go. Okay, so and what I'm going to do now is I'm going to centrifuge it again at a fairly low speed, just like before, to remove still some more of this solid milk protein. Okay. If you don't have a centrifuge, can you swing it around along a long rope? I know that there are those, I don't know if this is going to be actually fast enough. You might want to try the following. You might want to use a strain or a sieve or some kind of a cloth. Try to do the following. Take some of the kefir and maybe squeeze it through a cloth. Maybe this also works. Yeah. Another off topic, in my conversations with AI, the AI suggested to use carbonated water to slow down fast microorganisms. Does that really work? Okay. Not tried it out. I didn't try this out. I can imagine maybe it does work. Because the carbon dioxide, if there is little oxygen and a lot of carbon dioxide, then this slows down also the cell respiration. They have less energy. Yeah, there is a comment here. Es besteht kein Grund zum Schreien. There's no reason for me to scream into the microphone. Okay, evidently you are able to hear me well enough despite the running centrifuge here. Okay. So I would say probably the problem with the carbonated water would more be that there are then those CO2 bubbles forming under the microscope slide. So maybe that could be an issue. Okay. Methyl cell, yes, this is methyl cellulose. That is indeed something that can be bought. It's a powder. You mix it with water, and then you have a very highly viscous solution that also slows down microorganisms. Methyl cellulose. So let's see a little bit how this actually, let's slow it down a little bit, if this actually works. Okay. Yeah, you see actually over here, yeah, that there is now still some stuff here. What I'm going to try now is the following. I'm going to do this again, and now I'm going to spin it at high speed. Okay, let me see if I can have another tube here. And then let's hope that this also works. If not, let me see. Here we go. So you're able to see me better here. So I'm going to now do the following. I'm going to decant the whole thing again. I'm going to keep, in case I don't have anything, I'm going to keep this one over here. You see actually there's this white stuff here on the side, yeah, which could also be already some of the bacteria, but I'm going to keep this here on the side over here. I'm going to add again a little bit of water. Where did I put my water? Here it is. And I'm going to now spin it at high speed, hoping that I'm now able to collect any bacteria. It's a little bit cloudy still, so let's see if this works. Okay, let's now go to high speed, maximum speed. Okay. And let's wait for a couple of minutes. If I do not see a pellet, in other words, if I do not see this tiny thing on the bottom, then I'm going to use this one over here, and I'm going to see if this works better. Okay. And you know what I'm going to do in the meantime? Let's look at this. you see the wobbly thing? That's the vibrations of the table. Yeah. So, and again, this was from before. Yeah, this wobbly wave-like thing is because the vibration of the table. It's interesting. Yeah. You still are. Yeah, it's interesting. Let's see. I'm occasionally also reading. Yeah. Why are bacteria on the surface during centrifugation? They have mass too. Yeah, that's an interesting one. The reason is the following: the denser the substance, the more they are pulled down. So they have a mass, but that's why I was spinning it at a lower speed, so that the bacteria stay more in suspension, right? And right now I was spinning at full speed, so that hoping that the bacteria will also settle down now. Okay, it has to do a lot with the density of the things that you want to centrifuge. And luckily, those milk proteins that you see, all of those larger particles, they seem to be denser, and therefore they start to precipitate down. So what you can also try to do is if you let the yogurt or kefir stand, you're actually going to see that it will settle down, and that there is a more liquid, clear part at the top. Yeah. Yeah, I'm making them really, really sick. The interesting thing is the following: is that this has to do, I think, with the camera shutter speed a little bit. So of course the vibrations are very fast, but the camera shutter speed seems to be somehow in similar frequency. Therefore, it makes this wave-like motion. Yeah. Yeah. So I don't know if this is gonna be enough. Okay, I'm going to. I guess I'm just gonna give it a try. If I see a pellet, I'm happy. If I don't see a pellet, I'll have to use the other one. Look at this. Wow. So let's go back to the desk view here. So I'm really nervous. So this is the balance, and let's see if we are lucky here. And indeed, do you see a little bit? Of course not. Here it is. Okay, good. So I'm going to be using this here, and I hope that there are going to be plenty of bacteria in here. So this means I have to get rid of the centrifuge now. Okay. So I hope that, here we go. So I hope that this pellet over here contains mostly bacteria. And I carefully decant everything. Very good. And usually there's always a small drop of liquid left. And this is when I will now use my pipette. Okay, I'm gonna take a new tip. And where are my tips? And yeah. Centrifuge without a centrifuge. I mean, there are some homemade centrifuges for very small amounts. Or what you do is you simply wait and you allow it to settle as much as possible just using gravity. It's going to just take a long time. So you see I'm resuspending this whole thing here. This centrifuge was not too expensive. I think I paid around 70 euros for it. And it's good enough for my purposes. So I have now over here, this in here, and let's now give it a try. It's okay here, a tiny, every time when you want to use, you always use a very small amount. Okay. And of course a cover glass goes on top. And then of course I'll be also doing a little bit of staining as well. And so here we go. I hope there's only one cover glass. Yes. Here we go. And this goes now under the microscope again. Okay, so this was the one that I prepared before the live stream. Okay, so this is the one. I'm going to remove those now. And we're now going to check whether the one that I made right now actually works or doesn't work. It's focus, 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 focus. There seem to be too many of them actually. Yeah, I used way too much liquid, but actually it works. You know what? I have to do the following. I have to remove some of this liquid, or I just, it's too much, right? This is way too much, but I mean, you see that all of those tiny dots in the background, so they're kind of overlapping each other now. I have to do it again. I have to use much less of this. So I'm going to use a tiny amount. Let's quit again. Okay, a tiny amount. Okay, you have not seen this now, what I've done on the desk. So I just show it to you over here again. You see it's just a tiny drop to put on here now. Because bacteria are so small that if the layer is too thick, then they're going to start swimming up and down vertically, and then you always lose focus. So you really want to make sure they're really compressed flat. And so that was the one from before, same sample, much less. Hope that this works better. If we only find it, yeah, here they are. Okay, okay. Yep. Yeah, because now maybe I used too little of it, so now they're scattered around. Yep. But those tiny dots in the background, these are the bacteria. So you know what I'm going to do now is just, I want to get a higher magnification. So I'm going to switch over to the 60 times. So, and now I had a little too little. Okay, here we go. Yeah, but here that's a little bit better here. But at least I think you see that by doing this centrifugation first at slow speed, then at high speed, at least we're able to remove all of this other stuff. Yeah. But still, I'm not fully satisfied. Now I used a little too little maybe. Okay, let's check the other one again over here. Yeah, this was the one. Yeah, this was yeah, a little bit more. So let's have it here. Yeah, I think this is better. Yeah. Let's change this around a little bit. So this is now DIC. I'm going to now remove DIC. This is now regular bright field. Okay, you see it's, yeah, we're also able to see it with regular bright field. And the thing that I kind of like here is that we're able to actually see different types of cells of bacteria. You can actually see, for example, rod-shaped ones. If you look very carefully, some of them are in pairs, double, diplococci, yeah, and so on. Did I identify the drain bacteria from last week? No, I didn't. Yeah. That's the thing. Doing a bacterial actual identification is unfortunately pretty complex. I mean, I'd have to grow them in pure culture, and then I have to do a DNA analysis. I did that before already when I was at university. I did a DNA analysis of bacteria. Look at this here. Yeah, also in chains, some of them here. Yeah. So okay, this is actually better now. Again, maybe here. So, and this is now bright field. This here is now DIC, where you can change the colors around. And phase contrast, let's have a look. Okay, is, I have to change the filter again. Is this here, and phase contrast is, shows you the bacteria, of course, dark on a brighter background. Yeah. Yeah, so. What I would like to do now is the following. I would now like to stain it. Okay, so, but I think you get the idea that by doing this centrifugation, whoop, I nearly tipped over the water, where we removed most of this, of this milk protein. Yeah, and by spinning it at a higher speed, we were able to actually precipitate and pellet, get out the bacteria. So I was quite happy that this actually worked as well as it did. But I do want to show you a little bit of staining as well. Therefore I need another slide over here. Let me turn off the light here. Okay, and I'm going to take again here a small amount. One of the reasons why I'm also doing this because occasionally people actually they approach me and they ask me, okay, oh, they want to see bacteria under the microscope, and what do they have to do in order to grow bacteria? And I'm never really happy when people actually want to grow bacteria because you never know what you're actually growing. But I have no problem when people actually put kefir, yogurt, and so on under the microscope because basically these are safe bacteria. Yeah, by the way, I know some people already asked in the past if I not want to put some sourdough also under the microscope for bread making. That's also on my agenda. So if I want to add some of the stain, it's crystal violet. I found this works quite well. Then we have to be aware this is a very, very strong stain. And I have to take a very small amount. This is already maybe probably a little bit too much. I have to take a very small amount here. Yeah, and I'm just going to put it here on the side, maybe even less. Yeah, so I'm going to put this away and let's mix. Okay, let's mix. And again, of course, cover glass. Okay, let's see if this crystal violet makes bacteria also visible. Okay, so let's go again to the microscope view. So that's the phase contrast view from before. This is the one that I tried then later on because, okay, here we are. Because I felt that the other one had too many bacteria, then this one over here has too few. So I'm going to remove those again. Okay, and we're going to now look at the stained bacteria over here in bright field. This means that I have to basically go like this here. It's way too bright. We see already that there are some this blue stuff here. That's still some of the milk protein. Yeah, we go up, and we go up one more. Just a second. So we have to adjust the light. This is now bright field. We have to maybe close the condenser a little bit. Yep, and I think we can also see now the bacteria much better, and you can see that the bacteria have now taken on also a little bit some of the blue stain as well. Yeah, so we can see that it's possible to also make those bacteria visible even if you, under quotation marks, only have a bright field microscope. Yeah. But of course, we always have to be aware that when we add some stain or any other chemicals, then of course there is the very real possibility that the bacteria will actually be killed by the stain. Yeah, because the stains, they react. Yeah. And for the fun of it, okay, you know what I'm going to do now, because it is a, let me remove the slides here, because this is after all a, how do you say, a temporary mount. Let me put this away. I need to wipe off. I'm now going to add a little bit of immersion oil, and let's do some oil immersion microscopy of this stained specimen here. Okay, so just for your info, this is now a 60 times with a 60 times objective. Okay, so, and I'm going to now rotate in the 100 times objective without immersion oil. Here we go. Okay. This is without immersion oil, and it's quite blurry. I have to find the focus. So that's how it looks like, right? So now we see actually the bacteria bouncing around. They're bouncing around because of so-called Brownian motion. And yeah, without immersion oil. So what I'm doing right now is I'm going to add now some immersion oil, and I hope that we're able to see the bacteria a little bit better. Okay. So I need to find my ball with immersion oil. So I've now remembered the position. I'm lowering now the slide and the stage. I'm adding now, you have to check what I'm doing up there. I'm adding now a drop of immersion oil directly on the microscope slide here. Let's raise it again. Let's raise it again. And here we go. And I have to find the focus again. And here we go. Okay. And now we can actually see the Kefir bacteria much better. Yeah. And yeah. And I have to tell you, I am quite happy with the result. Okay. So when we are basically eating yogurt or kefir or any other fermented foods, and of course, we're eating a lot of bacteria as well. I mean, that's the whole point. Yeah. So I'm quite happy with the result, especially with the fact that it's possible to concentrate those bacteria using centrifugation. Okay. So, and what I'm going to do now is the following. I'm going to now check some of your comments. Okay. Yeah. Make water kefir and milk kefir. Do you believe that these are very healthy bacteria? Well, let's put it this way. Healthy and healthy. They are basically food that we're eating, and certainly they're not harmful. Otherwise, fermentation gone bad can make you pretty ill. But you'd probably also taste that it doesn't taste well. But those bacteria in kefir and also in other fermented foods are actually bacterial cultures that are known now and that basically are used now in food production. Yeah. Not all bacteria are stained. That is also a point. The important thing is the following. That's correct, and I think it is like this that I do see them a little bit better than without the stain, but it is also like this that if you, for example, defocus a little bit, then you're going to see that some of them actually look brighter than others, and this is the way that the light is refracted. So what we have over here is actually a combination of staining and also of light being refracted differently. Yeah. So this is essentially something that we have. We have basically two factors or parameters over each other. Yeah. Would gram stain make sense? Yeah, I could, not now because I don't have the time now, but this is indeed something that I wanted to do as well. That essentially, but lactobacillus and so on, actually they are mostly gram positive. Okay, so most of the, many, but not necessarily all of them, but they can be they're gram positive. While I listen to you, I'm filming some Vorticella from a rain puddle. What should I look for to try to identify them? Oh my gosh, there are so many different types of Vorticella. What I would recommend is, in any case, is to take a picture or a screenshot, document it, and then try to the best of your knowledge using an identification book. And what I've seen is that in many cases, even if the identification book contains many of these specimens, very often, they don't always look exactly like the ones that you find in a real water sample. Yeah. So Feusner, yes. Yeah. Feusner is Mr. Feusner. He was from the University of Salzburg. He was a protozoologist. And unfortunately, he passed away already a few years ago. But he made all of his publications available online. Okay, so Mr. Feusner is quite a well-known protozoologist. Yeah? Can you PVA glue them or heat fix? What's better? The question is are you talking about now the Vorticella or the bacteria? The bacteria generally, you can heat fix them, of course, but you normally do that, of course, for staining. And what you have to do is you just have to try out different mounting media, whichever one works best. PVA glue, I have not made any permanent mounts yet, so much of bacteria, because I always look at them fresh. Yeah. Can these bacteria be fed to microorganisms culture? Most bacteria should do, I believe. I see no of course you can do that. What you do is if you want to, don't overfeed them. If you, for example, want to grow paramecia or Vorticella, what you can do is you can make a so-called a milk culture, where you feed them either yogurt or milk, and then they will actually eat those. But you have to be extremely careful that you do not overfeed. That can happen very easily. You have to just at a tiny amount, just that the the water becomes slightly cloudy, and then you have to wait for a few days until the cloudiness disappears, and then you can add some more. Yeah. So, when are you going to update the list of Patreons on your site? Thank you for asking. This is actually something that I wanted to do. My website is completely being rebuilt right now. The thing is, the following, my website was down because it was under attack. And a few weeks ago, about 2 weeks ago, I started completely rebuilding it. So the templates, if you just visit the microbehunter.com website, it's a disaster. The articles are there, but it does not look very nicely formatted yet. And I'm currently in the process of completely redoing it, and I'm going to update also the Patreons on the website. Okay, but thank you for reminding me. I did not forget about this, but I was panicking a few weeks ago because my website was completely down. And luckily now with Cloudflare, it works again. Okay, so what I would like to do now is I would like to move on a little bit, and I would like to, I would like to completely change topics. I would like to talk a little bit about some microscope hardware, if it's okay for you, because some people actually wanted to know more about this, and I therefore brought along a couple of phase contrast condensers and so on, simply to show you a little bit of how this works. And we've already looked a little bit at phase contrast before, and I would like to show you now the optics behind the whole thing. Okay, so what I'm going to do is over here, I'm going to switch over to the desk view. And I'm going to also explain some of the struggles that I had in upgrading some microscopes to phase contrast. So look what I have here. I'm going to make this a little bit over here. Over here. This one over here. This one over here. And yeah. And two more. These are called Bertrand lenses, or phase telescopes, as they're called. These are special types of eyepieces. Look at this, what we've got here. Okay, so I need to explain this. Okay, so just for your information, this is for a traditional Olympus microscope. However, many other brands also looks very similar. And the purpose of phase contrast is that you're able to see very transparent objects like bacteria, like we've seen before, that you're able to see them dark on a brighter background. So in other words, the optics, they convert the differences in refractive index into differences in brightness. And a Danish physicist, Fritz Zernike, back in the day, he won the Nobel Prize for having invented this method. And it is still used, obviously, because it's very practical. Yeah. So Olympus BH2, it is compatible to Olympus BH2, but I've got this mounted on my Olympus CH40 and on the Olympus CHA. They're all compatible. Okay, so so that is something that I just want to make clear, and I want to tell you some of the struggles that I had with those. Okay, so what I've done is the following. I went on eBay, and I bought myself, for my Olympus microscope, I bought myself this objective here. PL stands for positive low. That means it's a phase contrast objective. All right. Achromatic 10 times, and I said, cool, now I have basically for my older Olympus microscope, I have now phase contrast, and I need a condenser, right? So I bought myself over eBay also this condenser over here, and look at the phase contrast microscope. That's really important. Do you see this ring in here, right? It's called it's called the phase ring. It's a semi-transparent, it's directly on the lens here, and this phase ring is extremely important, yeah. And what you need is for this objective, you need a condenser, a specific condenser, and that's the one that I bought, which also contains, specifically for that objective, you have to have a setting. Like over here, you have to turn this all the way until you also have 10 times, right? And then you will see over here, okay, where is this? Here, okay, you're actually going to see that this over here, this phase, it's called the phase annulus. So this kind of allows lights to go through in a ring shaped. Yeah, and this has to overlap exactly with this one over here, right? And what do you notice already? And this is something that I bought this here. It was not so cheap. I bought this here, was not so cheap. And I said, okay, now I can finally, both are from the same company, Olympus, the traditional 160 millimeter system. And then it did not work. Oh my gosh, why not? Look at this. Because the size of the phase ring and the phase annulus are different. Yeah, it's different. They don't match. And the reason is because they're for different generations, so to say. Yeah. So what I had to do therefore is I had to go out, shop again, and I found myself another one. This one over here. Okay. Over here I also have to find this here. This is a little bit more difficult to read now. Okay. Over here, I don't know where this is 40 times, so I think this here is 10 times. Yeah, here it says here. No, that's not 10 times. That's, let me see. That's dark field. That's 40 times. That's 10 times. Here we go, right? That's cool, right? So, and this one over here, do you see this one over here, right? This one here was actually matching, yeah? So basically, I got it to work, okay? So this is yeah, something I did not know, that essentially that even from the same company, there are actually two different generations of phase contrast. And the take-home message is that you cannot buy the condenser separately from the objectives. They have to match each other, right? So those two match. Now you're going to say, okay, what about this one over here? So how do this one over here, how does this actually look here? How does the objective for this one? It's actually a nice, high-quality metal one. Yeah, indeed. Look, do you see this here? This is also a traditional Olympus objective, also with a phase ring. And now this one here matches. Okay, they overlap, they match. Yeah. So yeah, different generations. So this is actually the older version, right? And if you now kind of wonder how can it be that this objective is so much smaller than this one over here? I mean, actually they have the same. Here they fit, but this is a short barrel objective. They basically were actually a centimeter shorter. So this has a conjugate distance of which is shorter. It's a short barrel objective. Yeah. So basically, Olympus over the years has also changed then the objective size. Yeah. You can also see from this must have been from the 1970s maybe. Huh? So this is kind of the way that it works. We have to match those two together. And now I want to show you a couple of pictures. Yeah. I want to show you. There's a comment about my book here. Okay, so this is what I would like to show you now. I want to show you a couple of pictures here, where no, it was a slideshow. Here it is. Yeah, and I'm going to So okay. I need to pause this a little bit. Just a second. Where can I pause this slideshow? Okay, I'm gonna pause this. Okay, nope. Here we go. If you look at this here, you can actually see that. Over here, you have to center the condenser. You see over here, this is the bright part here. That is from the condenser, the light. And you see here slightly on the side, not perfectly overlapping. That is the phase ring from the objective. Okay, and you see that it's not fully overlapping. That's why you have to center the condenser. Yeah, I'm going to show you also how this is done, centering of the condenser. Let's continue with the slideshow. How do I do this? Yeah, over here now it's centered. Ah, gosh, why does it always disappear? Okay, let's go to the next one here. Now you see it's up. Now you see it's centered. You see over here the white part from the condenser overlaps with the darker ring here. That's the way it should be. And when the condenser and the objective don't match, then one of them will be larger and the other one will be smaller. So you cannot overlap them. And this is the problem that I had. Yeah. So how did I make that image? Yeah, I'll show you how I made that image because it's the next thing. Yeah. So let's move on a little bit. Where's this? I need to go to the next one. Yeah, that's basically how it just looks like without. Yeah, that's just the ring from the objective. Yeah, here it's not aligned. Yeah, you have to move the condenser around to align it. And here it is aligned. Okay, so 2 questions. How did I make this picture? And how do you actually align the whole thing? Yeah, okay, so I'm going to show you both of these things here. Over here, let's have a look at this one over here, the different ways. Do you actually see this? Yeah, yeah. By moving this here, yeah. I don't know if you're able to see this, but it moves a little bit. It's barely. Yeah, I don't know where. Maybe you can see it here a little bit. Yeah, if you move it down, you see. I don't know. This is almost. It's so little. Yeah, here now you see it. Do you see? By shifting this, moving this back and forth, you see actually how it moves in one axis. Yeah. So this is how you can center it. Yeah, and you have to move it so until both of these rings overlap each other. Yeah. So that's this is in one dimension. This one is in the other dimension. Yeah. I don't know. Maybe I don't know where you can see this. Actually, it's so little that you can almost not see it. But that is here again. Yeah, you see how it's moving back and forth. Okay, for centering. And what about this one over here? This one over here can also be centered, but you have to use a screwdriver. Yeah, to center it. So these are the centering screws, and this also moves this around so until basically it's perfectly centered. Yeah. So that's another thing that you have to do, is you have to kind of adjust everything until everything's perfectly centered. Now the question is, how did I make those pictures with the overlapping images? And that's where you need called Bertrand lens or phase telescopes, they're called, right? So they're inserted into the eyepiece, and then you look through it, and then you can actually see. So these are not regular eyepieces, yeah, and then you can actually see how the 2 rings, if they're actually overlapping or not, and you have to focus this as well by turning it in and out until you get a sharp image, yeah. And that's basically what I've done, is I've basically inserted this into the eyepiece, and I put my mobile phone camera on here to take a picture, yeah? And you actually see it exactly the way that I showed it to you, yeah. Yeah. So that is the thing here, right? So yeah. What you therefore, if you're interested in phase contrast microscopy, just be aware it does cost a little bit of money, and it's not suitable for all specimens. Yeah, so only very thin, flat specimens are suitable. Okay, so yeah, so this is. And another thing, what I just want to mention is the following, and I've seen this online somewhere as well. What some people have done is indeed they bought just an objective. They did not buy the condenser, but they actually made 3D printed or something their own annulus. So they basically kind of did a little bit of trial and error until they got the correct size. This takes a lot of patience. Yeah, but some people actually, or they used some kind of a clear plastic film and kind of used a dark inkjet printer or a printer to print on the foil to block out the light. Yeah, so, so this is, I've been planning to try printing my own annuluses too. There is actually somewhere a website, somebody actually published some information. Was it Microscopy UK, maybe, who has actually done that? Yeah, it does take a little bit of patience here. Having said that, I want to do the following now. A couple of questions. Will this be in your book, and when will the book come out? Yes. So I need your help maybe. Yes, I am writing a book, a microscopy book, and I am bouncing back and forth, and I have not found my, direction yet. There are 2 options, and I would like to hear your view on the whole thing. Option number 1 is to do a more or less complete introductory microscopy book, including how to buy a microscope, some optics maybe, specimen preparation across the board. That's kind of idea number 1. This is going to be a slightly larger book. Maybe it's going to take a little more time. Or possibility number 2, and this idea has been floating now around in my head for 2 weeks now, is I reduce everything and I do just specimen preparation. So people have a microscope, and they basically don't know what to observe. And I would just like to do then a book just on explaining specimens and how to prepare them. What should I do? I'm just throwing the question out into the community. A more comprehensive book, or a book that basically is focused mostly on observation and specimen preparation. Just asking. Okay, when will it be available? I don't know, but I want to actually use the summer holidays, the upcoming summer holidays, to actually get most of it finished. As a matter of fact, I've got a significant part of the manuscript already finished. Yeah, so this is something. But I'd like to hear your view on the whole issue. Okay. Okay. So okay. Yeah, Microscopy UK. I like both ideas. Could do the basics first and do a second book. That's also what I've been thinking about. And I will reveal another little thing. Yeah. What I want to do, I would like to actually, for the different chapters, I would like to make also short accompanying videos. Basically with a QR code, which are only for the people who buy the book, where the things are also explained briefly in a video. Still thinking about it. Okay, so before I stop, I promised my students to show them something, okay? Because maybe there are a couple, many of you know I'm a teacher, and my students surprised me the other day, and I said, as a thank you, I'm going to feature it in the live stream. Okay, I got this here. And I would like to say, therefore, an official thank you to my students. It's a plant cell. And yeah, they spent 5 hours making the cake for me. And I was able to identify the nucleus, the endoplasmic reticulum, mitochondria. I suppose that this is supposed to be a vacuole, cell wall and cell membrane, a whole bunch of vesicles, and the green things here are the chloroplasts. Again, a thank you to my students. I promised them that I'm going to show a picture of their cake in the live stream. Okay, it was a great cake. I loved it, and maybe you can make another one for me, for us teachers. Okay, yeah. So I cannot show the faces of the students, but I kind of promised them I'm going to show it in the live stream as well. Okay, it was good. Yeah, it was a lemon taste. Yeah, so this is just something I wanted to show you as well. And I would like to I would like to simply ask you if you have any recommendations or wishes for the book. Yeah, there's one interesting comment here. There are already a lot of books about microscopes, but specimen preparation and staining is usually not covered beyond the onion cells. That's actually correct. I was thinking of doing, 101 specimens to look at under the microscope, and not only how to prepare them and how to, but also a little bit about the context and what it actually shows. So a little bit of explanation as well, so that the things that you see actually start to make sense. This is a little bit the idea that I have, right? So easier and more difficult specimens. Yeah. Did they get better scores because of it? Unfortunately, that's not possible. Only 101. I was thinking 99 or 100. I don't know. Yeah, so I would like to show again over here. Again, those bacteria are still moving around. Okay. And yeah, so yeah, and that's basically all I wanted to show you today. I'm going to quit. Yes, and I do have to announce the following. An electron microscope. Yeah, I do also have to say another thing. Yes, unfortunately, two short announcements. Unfortunately, next week I cannot do a live stream yet again because I'm again busy. And I do, however, now publish, maybe some of you have seen it, explainer videos on my other channel. So I'll called just Microbehunter. It's going to be the next video that I'm going to release is going to be about viruses. So it's going to be a little bit more biological, so less microscopy, but a little bit more biology and microbiology explainers. Do check out the other channel as well. And I know I've not been uploading other regular videos on this channel during the last couple of days, but I will do so in the future again. I was quite busy the last 2 weeks. Yeah. And yeah, just wanted to keep you informed, keep on checking. Off topic, how would you feel about a user creating a Microbehunter fan club Discord server? Honestly, I've been thinking, I have nothing against it, but I think it would only make sense if I'm also kind of contributing to it. I have to tell you, I've been I once registered a Discord server, but I'm a little bit too busy with all of my channels and book and live stream and so on, and newsletter. I want to publish a newsletter once a month, so I cannot yet do another, how do you say, another platform, because it simply run out of time. I do have a Reddit page as well, where I also post links. I don't do a lot of conversations there, but I also post links there as well. So if you want to share something on my Reddit page, so r/Microbehunter it's called, you'll find basically also links to the videos there. Yeah. But I'm currently, I don't mind if other people are starting a fan club, obviously, but I'd have to, if I want to start something like this, I have to think about it so that I actually have the time to administrate this. Yeah? Your knowledge is expansive, and oh, thank you. Yeah. I would like to see a cookbook style recipe book for techniques in your videos. Yeah, so this is actually something that I was actually thinking, a little bit cookbook style materials, methods, step by step, what you can see, why it's significant, yeah, why it's interesting, plus a link to a video on how it's actually done. Yeah. How can we share our pictures or movies in this live stream? This is actually a good point. Tube PKN. My suggestion is the following. I mean, one thing that you could do is the following. Why not? Let's do this. I will post a link to this live stream on my Reddit page. And on the Reddit page, you can write comments and you can also post things. Yeah, I think that would also be a possibility. Yeah, yeah. So because I know, unfortunately, it's not possible to share anything in a chat or in the comments in YouTube. Yeah, unfortunately. Yeah, but if you want to, please visit the Reddit page. It's not very active. I just have been so obviously not, because I've just been posting links there. But if you also want to start a community there, which where basically you exchange some ideas and pictures, then that's certainly something that we can also do. Yeah. Okay. Yeah. Okay, you know what? I'm going to next week. Unfortunately, I cannot do a live stream. Again, I will, however, try to upload a couple of regular videos. Okay, I'm going to call it a day for today. Okay, problems with the forum. Okay, sorry about the forum issue. Sometimes I'm getting so many emails that I'm not seeing that they kind of disappear a little bit. But what I will do is I can reset the password for you. Yeah. Okay. Yeah. Chess? No, not chess. Something else. Okay. But I'm going to call it a day for the day. Thank you that you joined me again. Yeah. I wish you all the best, and see you around again. And as always, happy microbe hunting as always. Bye bye.